Have you ever found yourself staring at a massive spreadsheet of genetic data, just completely overwhelmed, and wondering, okay, but what's the story here? Well, it's a huge challenge in biology. So today, we're going to dive into a really powerful method that's designed to do just that, to find the hidden narrative in your genes and turn all that complicated data into clear, understandable insights.
All right, let's set the scene a little. Picture this. You're a researcher. You've just wrapped up a huge, expensive gene expression experiment. You've crunched all the numbers, and you finally have your results. But what you're looking at is, well, this.
And there it is, a list of hundreds, maybe even thousands of so-called significant genes. It's just a wall of text, right? It doesn't actually tell you what's happening inside the cells. This is that classic moment in research where you just throw your hands up and say, now what?
But here's the secret. The breakthrough comes when you shift your whole perspective. You see, the story isn't in any single gene. It's in the teams they form. The real magic happens when you see how they cluster together into these functional groups, these biological pathways.
And this is where a really cool technique comes into play. It's called Enrichment Analysis, and it's basically a detective that's designed to find that hidden story buried deep inside your long, boring gene list.
So what is it exactly? Well, to put it simply, enrichment analysis takes your list of interesting genes and it compares it against these huge databases of known biological functions. It's basically asking one simple question. Is my list of genes popping up in any specific pathway way more often than you'd expect just by random chance?
Now I know, that might sound a little statistically heavy, but trust me, we can break down the entire idea with a super simple game of chance. Let's just imagine an urn, like a big vase, that's filled with color balls. This analogy is honestly a fantastic way to get your head around the whole process.
Because this is the fundamental question we're trying to answer. When you see a pattern in your data, how do you know if it's a real, meaningful biological signal, or if it's just a statistical fluke, just some random luck? This little game is going to show us exactly how we figure that out.
Okay, let's play. First, the urn itself represents every single gene you measured in your experiment, the whole shebang. Second, inside that urn, you've got balls of two different colors. Let's say white balls are your significant genes, and black balls are all the others. Third, you're going to reach in without looking and pull out a handful of balls. That handful represents all the genes known to be in one specific biological pathway. And finally, the big question. You open your hand. Did you end up with way more white balls than you should have, just by dumb luck?
So that's the game. Pretty simple, right? Now let's take that simple idea and connect the dots back to the actual biology, because every part of our little urn experiment has a direct counterpart in real-world genetic analysis.
Let's make this super clear. The urn, that's your gene universe, every single gene you measured. The white balls, those are the significant genes you found. And the black balls are, well, all the rest. And that handful you drew from the urn, that represents one specific gene group or pathway that you want to test.
And hey, for anyone who's curious about the math behind it, there's a fancy name for this. This whole drawing balls from an urn problem is modeled by something called the Hypergeometric distribution. It's just the precise statistical tool for calculating these kinds of probabilities. All right, enough theory. Let's see how this whole thing works with some actual numbers from a real study. I think this is where the concept is really going to click and become crystal clear.
Okay, let's build our scenario. In this particular study, the researchers measured a grand total of 21,989 genes. That massive number, that's our urn. That's every single ball, black or white, that's sloshing around inside.
Now, out of that giant pool, their initial analysis flagged 518 genes as significant. In our game, these are the valuable white balls that we're hoping to find. They're mixed in there with tens of thousands of other non-significant black balls. Next, the researchers decided to look at one specific biological process they were interested in. And they know from databases that this process involves a team of 18 specific genes. So, this is our draw. We're metaphorically reaching into that giant urn to pull out this specific handful of 18 balls. And here it is, the big reveal. When they checked those 18 genes in the pathway, they found that a whopping 14 of them were on their significant list. So, out of the 18 balls in our hand, 14 of them are white. I mean, that feels incredibly unlikely, doesn't it? And that leads us to the million-dollar question. If you just randomly scooped 18 genes out of that massive pool of almost 22,000, what are the odds? What is the actual probability that 14 of them would just happen to be from that tiny group of 518 significant ones?
The answer to that exact question comes in the form of a little number called a p-value. And all p-value is, really, is the calculated probability that what you saw, our 14 out of 18, could have happened by nothing more than random luck.
And the result in this case? It is staggering. The p-value is basically 0. This means the probability of this happening by chance is pretty much impossible. This isn't random. This isn't luck. This is a statistically significant discovery. So, we have this tiny p-value. What have we actually done here? Well, we've done something pretty incredible. We have gone from a meaningless list of data to a genuine biological discovery.
We proved our gene list isn't just a random collection of names. We showed it's strongly, significantly connected to a specific biological process. And that's huge, because it tells us what's actually going on biologically in our experiment. It turns that boring list of data into a real, compelling story.
Okay, now for a really important detail. Kind of a pro tip here. A single gene can be part of multiple pathways, right? This creates all these messy overlaps. So when you test hundreds of pathways at once, just by pure chance, a few are going to look significant, even when they're not. Because of this, the p-values you get are often unadjusted. The simple takeaway for you is this: to avoid getting fooled by randomness, you really only want to trust the very, very tiny p-values. The ones, like we just saw, that are practically 0.
So at the end of the day, enrichment analysis is really a tool for listening. It helps us hear the story that our data is trying to tell us all along, letting us move beyond just raw numbers to find those hidden biological narratives. So the only question left is, what story is waiting to be heard in your data?